Well, it's almost time to start, so let's do. It's been a long time. Maybe we should start with introductions. Oh, that was supposed to be a joke, so I'm not serious about that. But let me tell you what I'd like to do today. First, you should have all gotten an assignment from me. And you may be wondering what that's all about. Well, it's an attempt by me to help you prepare for the remainder of the exams that we have in this course. And I'd like you to ask yourself, as you go over the various topics, what is there that I don't know? And then compose a question asking about that topic that you don't know about. I don't want you to ask me or to pose questions that you already know the answers to. So it's what you don't know. And I'd like you to each submit 3 such questions worth 5 points each. So if you pose 3 good questions, you get 15 points. If you pose 2 good questions and 1 bad, you get 10 points. And who decides if it's good or bad? Me. If it's frivolous, then we won't consider it. Okay, so any questions about that assignment? It's due Friday at midnight. And so over the weekend, I will review the questions you submit and compile them and distribute the compilation to you. And then on Monday, next Monday, we'll go over the questions. We'll deal with as many of them as we can in the class period. And it's an experiment. We'll see if it's actually helpful for you in your studying and for mastering the information in the course. And as I indicated, the topics to be covered are the same ones that were covered on test 3. So any questions about any of that?
I have a question.
Uh-huh.
So like, what would you consider to be a bad question?
A bad question would be, why is the sky blue? Something that's either obvious, the answer is obvious, or it's irrelevant. But I don't think that should be a problem if you really follow the instruction to ask questions about within the topic, about things you don't know. And you can, as you go through the materials, and by that I mean the slide sets, the recordings, your notes, you should come to issues that you either don't know enough about, wonder about, or don't know anything about. Ask a question about it.
I have a question also.
Uh-huh.
So you said something about test 3. Do you mean we're only like asking questions about the topics we went over for test 3, or is it over the whole course?
The former, just the topics that were covered by test 3. And I think that starts with Mendelian genetics and molecular genetics and go from there. And it ends up with these 3 big interrelated topics: energy, enzymes, photosynthesis, and respiration. Those are all really important, and so there's a lot of emphasis on those topics in this course.
Does that mean the final will be mostly on the topics from test 3, or is it just like as a review?
The final, okay, let me just go over the schedule as I'm visualizing it right now. So today is today, and we're going to review the last quiz and the last test and finish up slides on respiration. And on Monday, we'll do the questions, your questions, and their answers. Then on Wednesday, I'd like to spend some time just thinking with you about energy production and climate change. That's a critical issue right now. And then spend some time on Wednesday reviewing cystic fibrosis. And this is to help you think about your presentations. Then the following 2 sessions I'd like to devote to your cystic fibrosis presentations. And I've decided, at least tentatively, to commit 2 class periods to that so that you each have more time to develop a presentation. So I'm thinking 3 or 4 minutes per presentation, but with the same issues being addressed. It's just that you'll have more time to elaborate and present illustrations. Okay, so that would be Monday, November 30th and Wednesday, December 2nd. And then the last day of the semester is Monday, December 7th. And on that day, I'd like to have test 4, which will cover everything toward the end of the course. And then your final exam would be a week later on Monday, December 14th. And my thinking about this is that you can really study for these 2 big exams together. And what you study to prepare for test 4 that will then be fresh in your mind for the final. So did you all get that? Write it down so you know how the rest of the semester is going to go. And if you have any comments about it or objections, speak up.
Amir is asking to be unmuted.
Asking to be unmuted.
Probably has a question or something.
Okay, I don't see that. Amir. Okay, Amir, I think you're unmuted. Can you say something? I don't hear anything from Amir, although his icon says he's unmuted. Okay, well, Amir, if you can hear me, but I can't hear you, type a message in chat. Okay, so much for the introductions. What I'd like to do now is share my screen, and let's go over questions and answers for quiz 8 and test 3. And
Professor,
yeah,
what days will we be presenting our presentations?
I haven't decided. I think probably what I'll do is just divide the class in half. On the alphabetical list, just divide the class in half, and it'll be about 15 presentations per day, and on 2 different days. And then within a group, I guess the best way for me to do it is just randomly call your name and have you do a presentation rather than do it alphabetically. Can you see quiz 8 on the screen?
Yes.
You can see it? Okay. Make it bigger. Okay, I'm going to ask for volunteers to do each of the questions. Who wants to do number 1? Which of the following statements are not true about enzymes? No volunteers? Somebody speak up.
I'll go.
Okay.
Veronica Munoz.
Okay, Veronica.
Okay, it's 1. Which of the following statements are not true about enzymes? A, most enzymes require companion molecules in order to be active. B, enzymes in general can tolerate boiling temperature without denaturation. C, an enzyme changes both the activation energy and the net energy of a reaction. D, an enzyme is to its substrate, a lock is to its key. E, a denatured enzyme can be restored by changing its environment. F, an enzyme structure is altered by participating in a reaction.
Okay, which are the false statements there? Well, let's take them one at a time. Do most enzymes require companion molecules in order to be active? Remember the terms cofactor, coenzyme?
So that is true.
That is true, and so you would not check it since we're looking for false statements. Okay, enzymes can tolerate boiling temperatures.
That is false.
Also, you would check that. And then an enzyme changes both the activation energy and the net energy of a reaction.
I believe that is true.
If you remember the graph showing the activation energy and the net energy, the activation energy is drastically changed by an enzyme. The net energy, in other words, the energy that comes out of the reaction, remains the same. So an enzyme does not change the net energy. It changes the activation energy, so that statement would be false. Okay, an enzyme is to its substrate as a lock is to its key.
That is true.
That is true, so you wouldn't check it. A denatured enzyme can be restored by changing its environment. In other words, is denaturation reversible?
No.
You're right, it's not reversible. Once you kill it, it's dead. So that would be checked as false. And then finally, do enzymes, their structures change by undergoing a reaction?
That is true.
Remember the definition of an enzyme. It's a catalyst. So it enters into a reaction without itself being changed. So that statement then would be false. Okay, any questions anyone have about the untrue statements there? Okay, let's go to the next one. We have a volunteer. James, you look like a good volunteer.
All right. Which of the following statements are true about enzymes? Enzymes raise the activation energy of a reaction. That is false. The enzyme's shape must fit the substrate's shape in order for the reaction to occur. That is true. An enzyme's cofactor is usually a metal. That is also true. The stomach enzyme pepsin has a pH optimum of 2. That is correct. Disulfide bonds are solely responsible for an enzyme's tertiary structure. That is true. And the rate of reaction is a function of pH. That is true.
Okay. Does anybody disagree with any of James's statements? What about E? Disulfide bonds are solely responsible for an enzyme's tertiary structure. James said,
Sorry, that's false.
yeah, you're right, that is false. If you recall, disulfide bonds are very important in tertiary structure because they're covalent and therefore strong. So they hold the enzyme together, but there are 3 other types of bonds, which you should remember, are also involved in tertiary structure. Okay, very good, thank you. Who wants to do the true and false, true or false?
I'll do it.
Okay, Jonathan?
Earth's sun is the only source of energy for the life on the planet. That's false.
Yes, you're right, that's false. It's the main source, but not the only source. Okay, next one. Life forms. Who wants to do that?
I knew that one. So I said life forms are either producers of captured energy or consumers of it. The producers include, and I said B and C.
Fungi and green plants? Anyone else have a comment about that? Well, green plants for sure. What color are fungi?
They're like, well, I mean, it depends, but tend to be like lighter colors.
Yeah, fungi do not have chlorophyll. And so, in fact, nothing else on that list has chlorophyll. And so the only producers on that list are green plants.
Okay, sounds good. Thank you, Professor.
All right, number 5. Volunteer?
I'll do it.
Santa, okay.
Photosynthesis and respiration have opposite metabolic functions, 2 sides of the same coin, so to speak. Photosynthesis is an anabolic process that makes glucose and oxygen from CO2 and H2O, whereas respiration releases the energy in sugars to produce carbon dioxide and water.
You got it right. So everybody note that down. I think that question came up again on the test in a slightly different form. But anyway, it's a very important overview of these 2 processes. Who wants to do 6?
I can do 6.
Okay, Isabella.
The capacity to do work is called energy.
Yes. Okay, very good. Number 7, the second law of thermodynamics.
I can read 7.
Okay.
According to the second law of thermodynamics, when all of the chemical reactions in a living organism go to equilibrium, the organism is I'm pretty sure I put F like when it was a question and it was wrong. So I'm gonna guess that it's A and E.
A and E, hyper-energized and manic. No, if you remember, to be alive, an organism needs a constant input of energy. And so if it's at equilibrium, it's neither giving off energy nor taking it in. And therefore, it would be
Dead.
Oh.
It would be dead, right? Yeah, the answers that people gave to this question were across the board. A few of you said dead, but most of the class indicated other things. Anyway, that's the answer. If it comes up again, remember, if it's at equilibrium, if there's no energy going in or out, can't be alive. Number 8. Nathan?
Number 8.
Yeah.
Okay. The electromagnetic spectrum consists of wavelengths ranging in size from a fraction of a millimeter to many kilometers. A small portion of this spectrum can be seen as a range of colors by animals as well as plants based on the absorption spectra of the photosynthetic pigment chlorophyll, explained why all plants are green. Chlorophyll absorbs red and blue wavelengths but reflects green, which is what we see when looking at a plant.
Yeah, you're mostly right. Chlorophyll doesn't absorb green. Red and blue are absorbed by chlorophyll, but because chlorophyll cannot absorb green, it reflects it, and that's why plants are green. So, Nathan, you're right. Number 9. Jada, can you do that?
Okay. Metabolic pathways that consume energy to synthesize, build up complex polymers from simple monomers are called anabolic. Alternatively, pathways that release energy by breaking bonds and polymers tearing down to yield monomers are called catabolic.
Yes, you are right. And again, 2 important terms to remember in your studies. Anabolic is to build things up. Catabolic tears them down. Anabolic consumes energy and ends up storing it. Catabolic releases energy from energy that's already stored in food, which is respiration. Okay, number 10, the answers are all given there. Feedback inhibition is where the pathway end product inhibits the first step of the pathway. Competitive inhibition, a substrate and inhibitor bind to the same active site, they compete for it. And if the inhibitor wins, then the reaction is inhibited. Reversible inhibition, the enzyme recovers when the inhibitor is removed. Non-competitive inhibition, the substrate inhibitor, they're both there, but they have different recognition sites. So the substrate can bind to its site, but the reaction doesn't go because the inhibitor is in its site. Let me back up. I said that wrong. The substrate cannot bind because when the inhibitor binds, it changes the shape of the enzyme. So the substrate no longer fits. So did I confuse you on that? On non-competitive inhibition, if the inhibitor binds to the enzyme, it changes its shape, and therefore it does not bind to the substrate. And E, the enzyme reaction rate, varies with temperature or the substrate concentration. They're all true. Okay, any questions about quiz 8? There is some overlap between quiz 8 and test 3, which we're going to do next. So I'm going to stop sharing this. Now do you see test 3?
Yes, we do.
Okay. Okay, there are 23 questions here. And let's go through them in the same way. I'll ask for volunteers to go through, to take charge of reading the question and giving an answer, and then we can discuss the answer. So who wants to do number 1? Life on Earth.
All right, I'll do it. In order for life to exist on planet Earth, life forms require a source of energy. Indicate the possible sources of energy for life on Earth. The correct answers are photosynthesis and chemosynthesis.
Yeah, that's right. All these other sources of energy, they are indeed sources of energy, but life forms can't take advantage of them. I put a false term in the list, phosphosynthesis, which fooled some of you. I think you confused it with photosynthesis, but it was meant as a non-answer. Okay, number 2. Who wants to do it? Which statements are true about oxidation reduction reactions? Oxidation reduction is a hard one, so we need a brave volunteer for this. Okay, Erica Berger, would you like to try? Okay,
I can try.
all right.
In oxidation reduction reactions, which statements are true? A, electrons are transferred back and forth between substances. B, transfer of electrons as asymmetrical. C, a substance that gains an electron is said to be reduced. D, only reduction occurs in photorespiration. E, when redox occurs anaerobically, there's a third-party participation. F, a substance that loses an electron is said to be oxidized. G, oxidation and reduction reactions always occurs in pairs. H, the function of redox in energy metabolism is the released energy for ATP synthesis. I say A, electrons are transferred back and forth between substances. B, a substance that gains an electron is said to be reduced. I also say G, oxidation and reduction reactions always occur in pairs. And H, the function of redox in energy metabolism, it's to release energy for ATP.
Uh-huh. All the things you said are true, but you missed one. Anybody notice the one she missed?
I think she missed, is it F, a substance that loses an electron is said to be oxidized?
Yeah, that's right. So good. Those are all statements about oxidation and reduction, which are critical. We'll go over a bit more about that later today. Okay, somebody for number 3. This is to interpret a graph. Isabel?
Yeah, I can try it.
Okay. Go ahead. Is that Veronica?
No, it's Isabel.
Okay, Isabel. All right. Can you read it?
Yes, so it says enzyme reaction rates are a function of temperature as illustrated in the graph. Among the statements below indicate which is true and which is false.
Okay, there are the statements and the graph.
Okay, so it says the slope
of the line at position 1 shows that the reaction rate is at equilibrium. Position 2 identifies the temperature optimum for this enzyme's reaction rate. The slope of the line at position 3 shows that high temperature stabilizes enzyme activity. Can't remember, but I think I put false for the first and then true for the second.
And the third?
Oh, and for the third, I also put true.
Okay. Well, if you look at the slopes of the lines, you're right about number 1. That's not a reaction at equilibrium because the rate keeps increasing as the temperature increases. Number 2, you're also correct. That's the temperature optimum. That's the temperature at which the reaction rate is the fastest. And then number 3, the reaction rate is going down as temperature increases. So what does that mean? It means that
it is decreasing?
Yeah, the rate of reaction is on the y-axis, and as temperature increases, the rate of reaction is going down, which means that temperature is, as it gets hotter, it's inhibiting the reaction, which means that the enzyme is being denatured. Okay, the next one is, it looks easy, but it's not. Who wants to take a stab at it?
I'll go. Veronica.
Uh-huh.
True or false? In the photosynthetic dark reaction, the Krebs cycle, aka the citric acid cycle, and the chloroplast stroma is responsible for ATP production. And that is, I believe, false.
Why is it false?
Because ATP, it's not just in the chloroplast stroma.
The dark reaction is in the chloroplast stroma, but that's not what's wrong with this question or this statement. Is it the Krebs cycle? Remember, there are 2 cycles that are important here that we're discussing in photosynthesis and respiration. And in the photosynthetic dark reaction, it's not the Krebs cycle, it's the Calvin cycle.
The Calvin cycle.
So yeah, a lot of people got this wrong. So note down that in the photosynthetic dark reaction, it's the Calvin cycle, and then where is the Krebs cycle important? It does the opposite. It's catabolic, takes glucose apart, and it occurs in the mitochondrion, not in the chloroplast. And we'll come to that, review it again later today. All right, let's move on. Matt,
sir, aren't there actually 2 things false in that statement?
Responsible for ATP. What are you thinking?
The dark reaction or the Calvin cycle is actually responsible for creating ATP or glucose rather.
Glucose instead of ATP. Yes, thank you, Scott, for pointing that out. Yeah, it makes glucose, not ATP. The Krebs cycle makes ATP. All right, number 10. Most of you got most of these right, but there were some significant wrong answers. So who wants to match up the terms with the definitions? Starting with the cross. What is a cross? From the list below. Anyone know what across is from that list?
I
Can
can
I
do this.
guess?
Sorry, Nathan, you go.
All right, no, it's fine. You can go. So is it the breeding of two plants or animals?
Yes, that's right. So what's an inbred? I actually,
Homology or homozygous?
yes, that's the answer that I was looking for. Now, many of you said an inbred is mating of an individual to itself. That's not really correct. A mating to itself is called a self. And it's not really an inbred, but it's confusing. And so people who said mating of an individual to itself is an inbred. Correct. Okay, what's a parental generation?
Oh wow. Kill me with the C1. He's gay for that.
Mute there, Mike.
Jonathan
I'll
Garcia.
see it.
The original individuals crossed?
Yeah, that's right. That's the parental generation. Then F1, that should be easy. Which one is it?
Oh wow. I think I died.
Who wants to say what an F1 is?
Progeny of a cross.
Right, and then an F2 is?
Progeny of a cross between two F1s.
That's right. Next two are easy. What's phenotype?
Phenotype's a pussy.
Phenotype is what?
Jonathan Garcia probably needs to be muted.
Okay,
Phenotype is the characteristics, visible characteristics.
yes. As opposed to genotype, which is? What is genotype?
Symbolic representation.
Yes. Okay, and a back cross?
Is it mating progeny to parent?
Yes. Yeah. It's as simple as that. It's just you take a child and mate it back to its parent. That's a back cross, and then a test cross is what?
To prove an individual's zygosity.
Right. You have an unknown. You don't know if it's homozygous or heterozygous. So you cross it to something that you know is homozygous recessive. And an allele.
Alternate form of a gene?
Yep, that's right. Okay, so much for that question. Let's move on to question 6. Another graph. So who wants to do this? There are 3 questions there based on the 2 graphs that are shown below. So let me read the first question from the graph on the left. Describe what happens to the enzyme's activity as its environment varies from pH 5 to pH 10. And this is an essay question. So I asked you to tell me what's happening at various points on this curve between pH 5 and pH 10. Who can describe that? Well, let me help. Many of you either didn't get this at all or only got it partially. So if you look to the left side of the curve, as enzyme activity is increasing as the pH increases. And some of you who started to get this right, got that much. But what you didn't say then was that at pH optimum, there's a peak of activity. And then as the pH continues to increase, activity goes down so that the enzyme is denaturing at this high pH. So that was something that, as I said, many of you didn't get right. I think almost everybody got the right-hand graph correct. From the graph on the right, indicate the color of the arrow pointing to increasing temperature causing increase in enzyme activity, that's the red arrow, and what's causing decreased activity, enzyme denaturation, that's the blue arrow. Okay, number 7, a true breeding tall plant. Someone give the genotypes and phenotypes of the F1, and someone else do the F2s. So what's the, let's think of the genotypes. What would be the genotype for the tall plant? It's true breeding, and it's dominant. Tall is dominant. So
2 capital T's.
Yes, so it's big T, big T. And so it's crossed with a true breeding short plant. So what's the genotype of the F1?
2 little T's.
Anyone else?
A big T and a little t.
Uh-huh. And the phenotype?
They are all tall.
Yes. Now, F2s, what are the genotypes? And there are 3 genotypes for the F2s. What are they?
Capital T, and then a capital T, a lowercase t, and 2 lowercase t's.
That's right. In a ratio of 1 to 2 to 1. And what are the phenotypes?
We have a 3-to-1 ratio of tall and short.
That's right. Okay, that was the answer.
Sir, if I could, a slight correction. You'll have 1 big T, 1 big T. You will have 1 small t, 1 small t. You will have 2 big T, little t, not just 1.
Did I or someone say that wrong? I hope that's what we had.
I believe that's what I heard, and I just wanted to make sure that that got out there correctly.
Yeah. Okay. Well, thanks. Yeah, what Scott said is true. All right. Next one. Pretty easy. I think almost everybody got this right. Who wants to choose the correct statement? Anyone?
I can do it.
Okay.
So should I just say the answer?
Yeah.
All right, so is it A, all enzymes are proteins, but not all proteins are enzymes?
That's right. And all the rest of the statements are nonsense. Number 9, another graph. And so the question is, in what body part or what type of environment would you expect to find each of these enzymes? So if you look at the relative reaction rate plotted against pH, you look at pepsin, the pH optimum is 2. And what body part do you find such a low pH?
In your stomach?
Yeah, to my knowledge, that's the only place that pH is that low. Okay, the red curve gives itself away, salivary, that means in your mouth, where the pH optimum is about neutral. And then arginase, I didn't expect you to really know where arginase is found in the body, but I did want you to say that its pH optimum is alkaline. So that's what I was looking for in that question. And the next one, color blindness. Who wants to do that?
I can do it. I have it written down.
Okay.
Let me just pull it out real quick.
First, tell us the chromosomes that are involved.
So for the chromosomes, I have X and Y, and the X has a star on it, which means that it has the trait. And then I have the other one as XX, and only one of the X has a star on it.
Yeah, and which is which, sex-wise?
So the X and the Y with the star on the X is the male, and the X and the X with the one star on the X is the female.
Uh-huh. Okay, and then the question doesn't ask for a Punnett square, it just says what are the likelihood that the children will be colorblind? So what's the answer?
50% of their children would be colorblind.
Uh-huh.
One being male, one being female.
Yeah, in other words, 50% of the males will be colorblind and 50% of the females will be colorblind.
Yes. And then one of the females is also a carrier.
Yeah, the one that isn't colorblind, that's right, is a carrier. So everybody understand that? There will be more questions about Mendelian inheritance on upcoming tests, so make sure that you know how to do them. Number 11, humans have been breeding living organisms for many thousands of years. And the technology used to do that is what? Employs fertilizing what with what? Anyone?
Egg and sperm.
That's right. So that's the technology that humans have been aware of up until recently, when genetic technology, recombinant DNA technology has been introduced. Okay, number 14, let me just answer this for you. Some of you were very succinct about this and answered it in just a couple of words. True breeding and F2. Several answers were, were that succinct, and I appreciate that because that really does answer the question. That's what Gregor Mendel's genius was, that he knew that before he started to cross plants with different traits, they needed, they needed to be true breeding. And so he inbred them for many generations before he started. And then he had the insight, I don't know why he did this, but he went to the F2. All right, the next one, indicate which items are true about chemiosmosis. Who wants to do that? Jose Jacques? Jose, can you do it? All right, then someone else volunteer. Fabian.
Yes, Professor. Which number 13?
Yeah.
Okay. So number 13 says chemosmosis is a critical step in ATP production. Indicate which items below are true about chemosmosis. So I've said 3 and what was it? 6.
Okay, anybody else? What about the first statement? It occurs during oxidative, but not photophosphorylation.
Isn't that false?
Yeah, that is false. The second statement. The second statement is the definition of chemiosmosis. Passing electrons across a semi-permeable membrane to generate ATP. Is it a catabolic process?
No.
No, it's the opposite. It's number 7. It is an anabolic. It makes ATP. Is it a source of electrons for the electron transport chain?
Yes.
Yes. And then which cycle is it a part of? That was sort of a trick way to say it. It's neither. It's not a part of either the Calvin cycle or the Krebs cycle, but related, directly related to both. All right, the next question, I wanted you to try to think your way through a problem. I put a lot more information in here than you really needed. But based on what we've discussed about chlorophyll and green pigments and the requirement for capturing light, and you see a bright orange plant growing always in association with green plants, in this case, gray-green chaparral. You don't see it anywhere else. You only see it growing on chaparral. So what do you think that might be? I had a lot of different kinds of answers, but what I wanted you to realize is that if it's always associated with another plant, and it itself is not green, that it's very likely a parasitic plant. Now, if you made an argument that it was a green plant, just happened to be there, and the green chlorophyll was masked by an orange pigment, I would accept that. I think only a couple of you made that argument, but the ones who got it right said it's a parasitic plant. Yeah, the next question is really a very important one. Somebody want to answer it? Products made by recombinant DNA technology are controversial, and the resolution to the controversy is that every product, whether it's produced with sperm and eggs or recombinant DNA, should be evaluated by the balance between its what and its what. I know you.
Benefits and the liabilities?
Yeah, the benefits and liabilities. If it's a good product, if it's more beneficial than it is dangerous, then fine, go ahead and use it. But these are judgments that have to be made by usually regulatory agencies. The next one, you have 4 possible terms there. Each term is used more than once. Scott, you want to take us through that?
So 16. Photosynthesis and respiration are complementary processes in which the products of one reaction are used as reactants in the opposite reaction. Fill in the blanks below, choose the following terms. Each term can be used more than once. So in photosynthesis, we have 6 molecules of CO2 plus 6 molecules of water produce 1 molecule of glucose and 6 molecules of O2. And in respiration, we have 1 molecule of glucose and 6 molecules of O2 are used to produce 6 molecules of CO2 and 6 molecules of water and energy.
Yeah, and I like this question because it encapsulates the opposing processes of these two terms, photosynthesis and respiration. One being catabolic and the other being anabolic. one consuming energy from the sun, the other releasing energy from the breaking of bonds in food. Okay, we're running short on time. Let's skip number 17. Number 18 is true or false. Denaturation results when the secondary structure of an enzyme is damaged. Scott and I had a discussion about this. The fact of the matter is that it can be a debatable answer. And so I gave credit for both true and false, depending on how you see it. The main components of a chloroplast, and I'll just tell you what they are. Chloroplast does not have a vacuole, has a thylakoid membrane, it has a stroma. Chloroplast is not a mitochondrion. They're two completely different organelles. Chloroplast does have grana. Does not have endoplasmic reticulum, has an outer membrane and a thylakoid compartment. So those are the answers for that. And number 20, you probably wondered, why was this question there? Not necessarily so much because of the coronavirus, but I wanted to give you an opportunity to see a question that your initial reaction is to throw up your hands and say, I don't know anything about this. So how could I be expected to answer it? But when that happens, in this quiz or this exam or anything in the future or outside of this course, you think you don't know something, just stop and ask yourself, is there anything that I know about this question? And what I was hoping that you would recall is that when we've talked about coronavirus in the past, we've talked about its structure, and we've emphasized that it has this spike protein, very important, critical component of the virus is a spike protein. And we also emphasized that the genome of coronavirus is RNA. So this virus is made primarily of those two polymers, protein and RNA. Well, we know that the vaccine is not protein. So what's left? RNA. So for me, the important part of answering this question is the process of asking yourself, what do I know? And by deduction, come to an answer. Anybody have a question about that, or why I put this question in? So we might have questions of this type in the future. And so if you see something, if you see a question and you say, this is completely beyond my comprehension, don't stop there. You ask yourself, is there anything I know about this? What is it that I know? And how can that lead me to a deduction? I think we'll stop there because I want to take the rest of the period talking about the other slide set. So I'm going to stop sharing this. And now you should see a PowerPoint slide overview of where we're going. If you don't see that, let me know. All right, this is again an attempt to remind you of how these terms are all interrelated. And we've gone through them all, and we're down to respiration. And I'm going to go quickly through the first part because we went through these before. I'm going to stop with this one just as a review. Now this is cellular respiration. And I'll give you a minute to collect your thoughts. Remember, that respiration starts with glucose. And the first thing that happens is glycolysis. And the result of glycolysis is the conversion of glucose to 2 molecules of pyruvate. That occurs in the cytoplasm. And while that occurs, 2 molecules of ATP are made. And that is substrate level phosphorylation. The pyruvate then leaves the cytosol, crosses the mitochondrial membrane. Inside, it's converted to acetyl-CoA, which is the first step in the Krebs cycle. And as the Krebs cycle spins, and we'll talk about all the steps in the Krebs cycle, 2 more ATPs are made. Actually, there are 4 made, but there's a cost of 2, so a net of only 2. And then electrons are generated, which go into the inner mitochondrial membrane, where the electron transport chain occurs. And so these colored balls here, the green, orange, the blue, and orange, represent the electron transport chain, which you remember is a downhill, stepwise process where electrons are transferred back and forth between carrier molecules. And at each step, energy is released, or an energized electron is released. And then you probably don't recognize this strange structure across the inner mitochondrial membrane. That is supposed to represent chemiosmosis, where the electrons that are generated in the membrane cross the membrane and generate ATP. And because there are so many electrons involved, a lot of ATP is generated, from 32 to 34 ATP molecules. So along with the 2 that are generated by substrate level phosphorylation, the total from 1 molecule of glucose is usually about 38 ATP molecules. I won't dwell on this, you saw it last time, and the only point here is that we say glucose undergoes glycolysis to give pyruvate. Well, the steps to do that are shown here in this slide. Acetyl-CoA, the structure, we talked about that last time. And we ended up with this slide. Go back. Acetyl-CoAX is a crossroads compound. It's a common pathway point for the extraction of energy from carbohydrates, fats, and proteins. Put those together, that is food. And so this is catabolic, but acetyl-CoA can also be used as a building block, and so in that sense, it can be an anabolic molecule. But right now, we're focusing on catabolic. So here you have the 3 main components of food: proteins, polysaccharides, and lipids. They can all be broken down into their monomers. Those monomers can be converted to acetyl-CoA, and that is the first step in the Krebs cycle, which itself generates ATP, but it also generates carrier molecules, electron carriers, which undergo oxidative phosphorylation and generate a lot of ATP. And these are the steps of the Krebs cycle. Circled are acetyl-CoA. It always starts with that, ends with oxalacetate. These are the intermediates. And it's indicated, first of all, you notice that a lot of NADH is generated. And there's one substrate level phosphorylation between succinyl-CoA and succinate, which yields 1 ATP molecule. All right, this slide tries to summarize everything that I've said so far about aerobic respiration. So you can follow it through. Start with glucose here at the top. That's the product of photosynthesis. Undergoes glycolysis, and in the process, it requires 2 ATPs. It generates 4 ATPs, so that there's a net of 2, and these are substrate level phosphorylations for a net of 2 ATPs. Then pyruvate can go from the cytoplasm into the mitochondrion, to the inner mitochondrial membrane, and there pyruvate can be converted to acetyl-CoA, and that is the first step in the Krebs cycle. Then you have these NADHs, which are generated, and the Krebs cycle itself generates 2 molecules of ATP, a substrate level phosphorylation. But the important part are these NADH molecules, and as well as FADH. And these go into the electron transport chain, and that is in the inner mitochondrial membrane. And you have this stepwise transfer of electrons back and forth. And at each of those steps, energy is released, and the way it is through chemoosmosis, which again, this little structure here across the membrane represents chemoosmosis. So electrons that are freed are transported across the membrane, and as they are, ATP is made. And it's this process in the inner mitochondrial membrane which is the most productive in generating ATP. So the 32 ATPs that are made by that, along with the 4 ATPs from glycolysis and the Krebs cycle, give you a total of 38 ATPs for each molecule of glucose. So this is something you should have in your mind, the image of these steps in your mind, because it'll help you answer a lot of questions. All right, this is a review, but to make it real, we start with pizza. Think about what pizza is made of. Well, cheese. Almost all pizzas have cheese. What is cheese? It's fat. So you have fat, carbohydrates. Where are those? Well, you have dough, pizza dough, mostly carbohydrate, and protein. Some pizzas have meat on them, others don't. Anyway, all the major components of food are found in pizza. And again, this is a review, but the energy in pizza is broken down through glycolysis to acetyl-CoA, which goes into the Krebs cycle, and then that produces energized electrons, which go into the electron transport chain and generate a lot of ATP. Again, this is a review. Now, fermentation, I'm going to skip a couple of slides and show you this one because it contrasts alcohol fermentation with lactate fermentation. When the environmental conditions become anaerobic without oxygen, if you're dealing with microbes, in particular yeasts, the process of glycolysis produces ethanol. And it does so by undergoing glycolysis to produce pyruvate. Pyruvate is converted to acetaldehyde, and that is converted into ethanol, which is a very useful product. Now, in muscle, the same process leaves out the acetaldehyde, and pyruvate goes directly to lactate, lactic acid. And that becomes important. You've all experienced this. Remember your muscles burning when you climbed 20 flights of stairs? This is why. Glucose with glycolysis is converted to lactate, which is a burning sensation in your muscle. And this happens because in high intensity exercise, blood flow to muscles is reduced, and inadequate oxygen in the muscles, which is anaerobiosis, and the muscles need ATP, and they need it fast. So they can either get a little bit of ATP from anaerobic glycolysis, or they can wait until their muscles become oxygenated and get a lot of ATP. Well, the muscles can't wait. And so they undergo anaerobic glycolysis, which produces lactic acid. That's why the muscles burn. And it's thought that this burning is a protective mechanism to prevent muscles from damage due to overuse. So this is something you can all relate to because you've all had this feeling and now you know why. The rest of the slides in this set are largely reviews. They're just presenting the same information in different ways. So on this slide, for example, again, you start with glucose, glycolysis, pyruvic acid in the cytoplasm. Pyruvic acid, pyruvate, goes into the mitochondrion, to the Krebs cycle. You get ATP, substrate level phosphorylation and glycolysis. And in the Krebs cycle, you get a lot of ATP from the electron transport chain, oxidative phosphorylation. And again, the stoichiometry of this we went over in previous slides. So a review stated in words: glycolysis is an exergonic series of reactions, which means it's catabolic. It occurs in the cytoplasm outside of the mitochondrion. It begins with glucose, and glucose ends up being broken into 2 molecules of pyruvate. And the cell makes a small amount, 2 molecules of ATP during glycolysis. Okay, this is more review. This is glycolysis broken into 2 sections, the next 2 slides. Again, you start with glucose, and here you can see where energy is used. This is an anabolic process, or a catabolic process. To go from glucose to glucose 6-phosphate, a molecule of ATP is used, and you get the glucose phosphorylated, and you end up with ADP. That happens again in the step between fructose 6-phosphate and fructose 1,6-biphosphate. Again, a phosphate group is added, and so it takes energy to do that step. But then the rest of the pathway is on this slide, and here's where you see phosphate entering, and you get NADH. You get substrate level phosphorylation, 2 more ATPs here. And so on the right here, I've indicated the glycolysis stoichiometry. It produces 4 ATPs. It costs 2 for a net of 2. And these come from substrate level phosphorylation. This is review. More review. It says the same thing that we've already discussed. And that's the end. And I have used all the time. I'm going to stop sharing. Any questions before we end the session? You know what we're going to do on Monday. We're going to discuss your questions. And you're going to submit your questions before midnight this coming Friday. Okay, no questions? Then I'm going to